Alright, welcome to my video on nucleic acids. This is chapter 19, and this is really kind of the last bit of information we're going to talk about. So I'm going to take you through this. I'm going to highlight a couple of important details so that you know for the final exam, okay? And I'm going to try to go through this in a fairly quick fashion so that you actually pay attention. Alright, so when we talk about nucleic acids, right, we know that these are the molecules of heredity, okay? So one of the questions that should arise based on everything we've talked about in this course so far is how do we actually know how cells actually know when to produce different proteins, right? And out of all the different possible amino acid sequences, how do they determine what amino acids need to be put together one by one to make these proteins that are going to carry out metabolic processes and help sustain life? Well, when we think about where this information is stored, it really comes back to 19th century biologists, which really helped identify that this genetic information, these molecules of heredity, are actually stored in chromosomes. Okay, and so this hereditary information was thought to reside in genes within the chromosome. And so if we kind of look at this picture over on the right, we can see that we have our chromosome, and if we kind of zoom in and zoom in even closer, we start to see what looks like these beads on a string. Well, the beads are our proteins, and wrapped around that is our DNA. So we have really long strands of DNA that is packaged into our chromosomes by basically wrapping it around these proteins called histones, which allow us to know which information to produce and when. So these DNA molecules, or the nucleic acids, were discovered back in 1869 by Friedrich Miescher. Okay, and basically what he did is he had a theory that information was kept in the nuclei, and so he treated the nuclei with acid. Okay, and he found that you got a precipitate that came out, and this precipitate contained both carbon, nitrogen, oxygen, and phosphorus. Okay, and so what he determined was that this nucleic acid was actually stored in the nuclei, and it was acidic, so it was nucleic acids. That's how he came up with the name. Well, we really have to turn back to that central dogma of molecular biology, because do we say that all the information of what proteins to produce and what their actual sequence is determined by DNA? Well, we know the central dogma has DNA is transcribed to RNA, and RNA is then translated to the protein sequence. Sorry about that. So when we talk about nucleic acids, there's two types of nucleic acids, the ribonucleic acids and the deoxyribonucleic acids, so RNA and DNA. Whenever we talk about RNA and DNA, it's composed of basically three parts. Okay? We have a sugar, a base, and a phosphate. Okay? And this is what we could term a nucleotide. So the nucleotide is going to have the phosphate, it's going to have a sugar, and it's going to have a base as well. Okay? Alright, so first we can start with the sugar. Okay, and the sugar here in RNA is going to be D-ribose. So we've talked about ribose, okay, and basically we can form the furanose form of D-ribose. On DNA, it's deoxyribose, which means that we've now lost an oxygen on carbon 2, so we have the deoxy form of deoxyribose. Okay, and so really the only difference between RNA and DNA here is looking at the 2' carbon, whether it's hydroxyl or hydrogen. Okay, and so this small difference is actually going to affect both secondary structure and stability. Okay? The most stable conformation is when it's in the furanose ring, and so we always draw DNA or RNA with the furanose ring. Alright, next we can talk about the bases. Okay, so the bases can be broken down into either pyrimidines or the purines. Okay, and so pyrimidine is just a single ring that contains 4 carbons and 2 nitrogen atoms. Okay. The purine is basically a pyrimidine ring with an imidazole basically fused to it. Okay. What you'll notice are both are unsaturated with conjugated double bonds. Okay. The purines are going to bond to the sugar of a nucleotide with this nitrogen 9 atom, and the pyrimidines bond to that sugar through the nitrogen, or the first nitrogen. Okay. So these are all going to be very planar compounds. They absorb UV light. Okay, they're weak bases and they're relatively insoluble in water, but once they're coupled to nucleotides or to the overall sugar and phosphate, that's going to make them more soluble. Okay. And so when we break it down the purines, we have two, which are adenine or guanine, while the pyrimidines are cytosine, uracil, and thymine. Okay? You do need to know these structures. I will not have you draw them, but you do need to be able to recognize the difference and identify which is adenine, which is guanine, which is thymine, which is uracil, and which is cytosine. Okay? So uracil is found in RNA, while thymine is found in DNA. Well, each one of these bases participate in hydrogen bonding. They have both hydrogen bond donors and they have hydrogen bond acceptors depending on the groups that are attached to it. So whether we have deoxyadenosine or adenosine, which means that we're either the ribo form or the deoxyribo form, okay, where we would attach to this base, so our sugar is R, we can see that we can highlight both donors and acceptors. Okay, and so here we have this NH2 group. Well, those hydrogens connect as a hydrogen bond donor, while the nitrogens in the ring connect as hydrogen bond acceptors. And you can see this for each of these four different bases. So, one of the things we need to talk about is terminology. Well, first we can talk about nucleosides. A nucleoside is a compound that consists of ribose or deoxyribose, which is bonded to the purine or pyrimidine base by a beta-N glycosidic bond. So, in our figure here, we have uracil, which is connected through a beta-N glycosidic bond to our ribose ring. Okay? Well, the ribose ring, we have carbon 1, which is our anomeric carbon. Okay? We number the sugars as prime. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime responds to, or is really a nomenclature for the carbons of the sugar ring, okay? Whereas 1, 2, 3, 4, 5, 6 would pertain to the base. Okay? So the carbon of that glycosidic bond is an anomeric carbon. And lastly, you can kind of see how this structure ends up being perpendicular, where the base is perpendicular to the sugar. So when we have nucleosides, which just means we have the sugar and the base, okay, no phosphates, well, we can name nucleosides by basically just adding the ending ID-INE to the root name of the pyrimidines, or osine, O-S-I-N-E, to the root name of the purines. And so we get things like uridine from uracil, okay? We have adenine to adenosine. So the O-S-I-N-E ending tells us that we have a nucleoside, which means that we have an adenine base attached to the ribose sugar. And this is adenine is over here. So, sorry, these should be switched. All right, so next we can talk about the conformation. That conformation could be syn or anti. Anti is what predominates in nucleic acids, which means that the base is kind of turned outwards and not exactly perpendicular over the sugar. Okay, so here you can see with syn, the base is directly above the sugar. With anti, that base is shifted out to the right. Okay. Other things to note, we use the abbreviations U, A, T, C, and G for these nucleosides, or we can add a D in front, which tells us you have the deoxy form. Okay. All right, so next we have nucleotides. Okay, and with the nucleotides, if I can get to that, means that we basically have a nucleoside, which is basically attached to some kind of phosphate. Okay, so we have a phosphoryl group that's esterified to the hydroxyl of the monosaccharide. Okay, most commonly we either see this at the 3' or the 5' hydroxyl group. So in our figure here in yellow, we have a ribose ring. Okay, we number those carbons 1', 2', 3', 4', and 5'. In blue could be the different bases, whether it's a purine or pyrimidine. That's linked through a glycosidic bond. And if we look at the sugar, we either have ribose with a 2' hydroxyl, or we have deoxyribose with a 2' hydrogen. Most commonly, okay, we basically have off the 5' hydroxyl, we have phosphates, okay, or these phosphoryl groups. And we can either have 1, 2, or 3. 3 of them means that we have a nucleoside triphosphate, okay, like ATP or GTP and so forth. We could also have the nucleoside diphosphate, which means we've hydrolyzed off one of the phosphoryl groups. Or we have what we just call simply a nucleotide. The nucleotide means that we have a single phosphate. It could also be called a nucleoside monophosphate. Okay? So if you see the ending nucleotide, the TIDE ending, means that you have a phosphate attached somewhere. Okay? If it doesn't say, then it's always going to be at the 5'. Okay? Otherwise, it would say 3' or so forth. All right, so when we talk about these nucleotides, we have the nucleoside 5' triphosphates. Okay, a lot of these are carriers of energy, like we've talked about before. Okay, they all serve as recognition units. Okay, and so here we have our different bases, the nucleoside nomenclature, and then the nucleotide. Depending on here, we have all monophosphates. It could be diphosphates or triphosphates. And with RNA, we see the same thing. Okay, so this is just a nice table with all the different nomenclature. Well, we talked about ATP. It's really kind of important in energy metabolism, as well as UTP, which is involved in gluconeogenesis. We have CTP, which drives lipid synthesis, and GTP, which is more involved in protein synthesis. All right, so when we talk about nucleic acids are the building blocks, or are composed of the building blocks, which are these nucleotide monomers. Okay, and so each one of th' these nucleotide monomers, whether it's guanine, adenine, and so forth, the nucleotide version, okay, means that they're going to be joined together by a 3' to 5' phosphodiester linkage. These form these nucleic acid polymers. Well, James Watson and Francis Crick back in 1953, basically won a Nobel Prize many years later for determining the structure of DNA and RNA. So when they looked at DNA, they found that they could look at the structure of the nucleoside, look at X-ray diffraction patterns, and they found that it formed this sort of helix structure. Well, another guy came along, Chargaff, and he basically found that if you took one of these cells from species and ground up the nuclei, that really the concentration or the percent of adenine and thymine was equal, and that the amount of guanine and cytosine were equal as well. And so what he came up with was Chargaff's rule, that basically DNA was a double strand, and that bases on one strand paired specifically with bases on the other strand. Like I said, Francis Crick and James Watson both won the Nobel Prize, and they've actually sold both of them now at auction for, I guess Francis Crick got $2 million. I think James Watson just recently got almost $4 million, or his family did. All right, so the last thing I'm going to talk about is just the basic structure of nucleic acids. So we know DNA is the storehouse of all biological information. That then is passed on to the RNA, which ultimately helps determine the protein that's being produced. So proteins and enzymes actually bind to DNA. They recognize certain structural features. They read that nucleotide sequence. And we're going to talk about next class really why that's important and how these different proteins may bind and what they actually do. Well, the primary structure of proteins we talked about was the amino acid sequence, And we connected those different amino acids by peptide bonds. Well, for nucleic acids, we basically have the nucleotide sequence, so which nucleotides connected to which, and each one of those are connected by a 3' to 5' phosphodiester linkage. So a peptide bond is just like a 3' to 5' phosphodiester linkage, where we're connecting these monomers together. So on the right, we can see that we have basically these different nucleotides of A, T, C, and G, which is determined by the base that's attached to our ribose or deoxyribose sugar. So here we see we have ribose because we have this 2' hydroxyl. We write this at 5' to 3', where we have a 5' N phosphate and this 3' N hydroxyl. All right, so if we look at this backbone, the backbone contains this 5' phosphoryl group. So you have a 5' carbon, 4' carbon, 3' carbon, and then you're going to have the phosphoryl group of the next phosphate. Now this oxygen here comes from the 3' oxygen of each ribose or deoxyribose. You'll notice the backbone is an extended conformation, so DNA is long and thin. And we always read DNA with structural abbreviations of 5' to 3'. Just like amino acids or proteins, we talk about N terminus to the C terminus. For DNA, we talk about the 5' to 3' end. You'll also notice that this backbone, is actually very negatively charged because we have this phosphoryl groups. Well, we need to be able to stabilize that backbone, and so usually some kind of positively charged proteins or amino acids like lysine or arginine may bind to that, as well as some small cations like magnesium. That's going to end it for this recording, and I will see you in class either Monday or Wednesday, depending on when you watch this.